King Conservation District. And I was a project manager for the Discovery Farms project we will be discussing today and sharing the initial results. We were evaluating the differences between different dry manure storage practices, both at protecting water quality, but also at producing a high quality compost product that was rich in nutrients and free from pathogens and weed seeds. And Zen, do you want to introduce yourself?
Yeah, so my name is Zen Wilhelm. I am the Conservation Research Specialist at the Whatcom Conservation District. And I got the opportunity to help with this project in terms of designing the research design and as well as a bit of the field work and data analysis. I'll be going over some of the results of that data analysis today during the presentation.
And I am living in a remote area. It's been a little stormy, so just to make sure I have a good connection during the presentation, I'm going to be turning off my video while I'm presenting and then turning it back on during questions. So to begin with, I wanted to chat about what exactly a Discovery Farms project is. Discovery Farms is a national research program based in Wisconsin, and it develops on-farm and related research to determine the economic and environmental effects of agricultural practices on producers. Washington actually had its own Discovery Farms program that was based out of Whatcom Conservation for several years. And that's where we started this project. So the National Discovery Farms program, it was formed on the premise that oftentimes research on farm best practices are designed and implemented by academics and agency professionals. Because of this, the outcomes of this research often don't reach the farmers and producers who would benefit from this knowledge, and the designs and proposed solutions resulting from this research, they can sometimes be impractical for farmers to actually incorporate into their management plans because of that disconnect and them helping to design the research project to reflect the realities of a working farm. And so this overall can make research less valuable. So a Discovery Farm project addresses this through supporting farmer-led research that occurs on real working farms to address research that is important and impactful both to the farmers and environmental professionals alike. Each project engages the help of a steering committee. This is comprised of local farmers that help design and provide input into all aspects of the project, from study design, research parameters, and outreach and education. And this Discovery Farms project was funded through a grant by the King County Wastewater Division. They have a Waterworks grant program. And before we begin, I would also like to take this time to give a really big thank you to our producers at our study farm for participating in this project. We could not have done it without them, as well as King County that allowed us to utilize one of their farms as a study site for this research. So just a brief overview of what I'm going to be discussing today. Some common issues that producers face with dry manure management. I'm going to go over some benefits of composting, and I'm going to review the study design. So that includes the research variables and project setup. And then Zen will be going over our initial research results with you after that. So the project is really split into two components. The first is the research, which is 2 years of sampling analysis and data. And the other part is education and outreach. This consists of farm tours, webinars, and this presentation is a part of that. So what are the expected benefits of this project? First is understanding the effectiveness of different dry manure storage options. So as technical service providers, we can provide valuable information to local farmers who struggle with dry manure management. The study will also promote natural resource conservation with a focus on water quality. So helping producers become aware of our services as a conservation district, helping to promote the adoption of best management practices. And this project occurred in a high priority watershed with a large number of our goal is to share this information with them, as many of them as possible, and to have local data that reflects the conditions that they work in. So this image is a pile of manure that is a common sight throughout King County, and it's one that many livestock producers struggle to manage. Manure management practices are also one of the most commonly prescribed best management practices by conservation district staff, as well as one of the most commonly funded practices that we cost share on at KCD. So if you picture this pile of manure on bare ground during heavy rain or a stormwater event, especially in the rain season when piles tend to get saturated, the stormwater will enter the pile, and as it moves through and alongside the pile, it's picking up all kinds of stuff, sediments, harmful bacteria, and valuable nutrients for plant growth that we actually want to retain in that pile. And as it exits the pile, it carries these pollutants into our streams and groundwater, which can be bad and have implications for both wildlife habitat and health and human health concerns. To help prevent this, KCD typically recommends storing manure in a bin with a concrete pad with walls and a cover over the manure. That's really our gold standard to both protect surface and groundwater and help produce a better compost product for producers to spread on their fields. However, we also know that there are barriers to installing this gold standard. It can be extremely costly. It can take time to install. It can require skilled labor that can be difficult to find, as well as specialized equipment. And so there are groups of farmers that would like to install this bin, but are unable to because of these barriers. And so this Discovery Farms project is evaluating how effective different dry manure storage practices that come at various levels of affordability or cost to install are at protecting water quality and also improving the quality of the compost. And our goal in this is to be able to recommend to producers, if you can't afford the gold standard, here's maybe another solution, depending on your individual circumstances, that could be a little bit cheaper and work for you. So this image is just briefly reviewing the pollution pathways that manure leachate and contaminants from manure and how they might get into our stormwater, from stormwater into our surface and groundwater. So you can see in the farm here, there is manure and animal and agricultural waste. There's also other pollutants like pesticides, fertilizers, and septic tanks. And so that stormwater can go down and over the ground and into our rivers, but it also can saturate into our grounds, even into our water tables, potentially our wells. And this is one of the main reasons we're so concerned about these pollutants in the Puget Sound. So it's not a surprise to many of you that it rains a lot here in Western Washington. And so this project took special consideration for our unique climate in the Puget Sound region. We have milder winters and significantly more precipitation than other areas of the United States. As you can see in this red circle, this shows we receive among the highest annual precipitation levels in the nation. We also receive the majority of our precipitation during winter months, and this is a time when farmers are collecting and storing more manure. So this increase in stormwater has implications for the amount of potential nutrients and pollutants being leached from a compost pile. There really hasn't been a lot of research done in areas like the Puget Sound with extended season of stormwater events on the effectiveness between these different types of dry manure storage practices, which is another reason we wanted to perform this research, to have data that supported technical assistance and recommendations we're giving to producers to assist them in making decisions on how to best manage their manure on a local level. So aside from decreasing pollution, composting has many benefits for farmers. Composted manure naturally contains nutrients that are important for plant growth, such as nitrogen and phosphorus. And when you add it back onto pastures or crop fields or even in gardens, it increases the amount of organic material This can lead to improved soil stability, reduction in compaction, improved water infiltration and soil water holding capacity. It also can improve soil biological properties such as microbial diversity and disease suppression, and also can improve nutrient cycling. So all of these are really essential for plant growth and vitality, but it can also save you a lot of money. So the nutrients provided by compost, or at least a high quality compost, can decrease the needs for costly soil amendments. The composting process also decreases the presence of weed seeds, so this can help ensure that more desirable plant species grow in your fields, and it can also reduce your need for herbicides. Many producers are also able to sell their compost for a profit. It can also be an essential part of maintaining and improving animal, livestock, and health. It reduces exposure to parasites and harmful bacteria, both from regularly removing manure from the confinement area or heavy use area during that winter rainy season, and also treating the compost before it's spread back into pastures where animals might graze. So throughout this project, we had a steering committee consisting of local livestock managers that helped us form our research design, and they helped us select the variables to test for that were important for them. And they also worked with us as technical assistance providers to select ones that were important for us for monitoring the pollution issues you mentioned earlier. So I split this out into three main categories. So there's composting variables. These are variables that ensured that the manure pile had all the needed ingredients to compost correctly, and it wasn't being impacted by anything other than the different storage practice types. We also tested between different manure storage practices, both in what was leaching off the pile and what was in the final product of the compost. The other category is nutrients, and these are both essential for plant growth and vitality, but in excess of these nutrients that we tested for, this can cause issues for human health, water quality, and wildlife. And we also finally tested for pathogens that are commonly found in manure and that are another major cause for concern in water quality. So for the composting variables, micro and macroorganisms like bacteria, fungi, some insects, these are the things that are largely responsible for transforming your manure into that high quality compost product. I like to think of compost piles kind of like habitats that require different elements for these organisms to survive and be effective. And so we kept track of these essential elements to make sure that the different treatment piles for the different types of practices had everything it needed to compost correctly, and that a lack of any of these elements wasn't interfering in the data results that we saw. So the first thing we were keeping track of was the carbon to nitrogen ratio. Many of you guys may know this as your browns and your greens. For this project, the browns primarily consisted of bedding and hay, while the greens, or the nitrogen source, primarily consisted of animal manure. So carbon, or your browns, this is what gives food or energy for these organisms to break down the compost, to reproduce, and generate the heat needed to kill weed seeds and pathogens you want to eliminate from your pile. Example of browns might be hay or unsullied bedding, leaves, wood shavings, or cardboard twigs and leaves. Carbon sources are also often an important source of structure to the pile. So if you have twigs or leaves or just anything that's a little bit larger, but not too large, you want it to break down, that creates spaces for airflow and oxygen to move throughout your pile, which is another important component of composting. Nitrogen, or your greens, this is what gives the building blocks for these organisms to reproduce. And so greens can include things like grass clippings, coffee grounds, and manure, or urine-soaked bedding. And so what can happen is if you have too little nitrogen and too much carbon, the compost pile cools and the process slows because there isn't enough of these building blocks for the microorganisms to reproduce and to effectively break down the different elements in your compost. If you have too much nitrogen, the compost pile gets too hot too quickly and doesn't have the energy to sustain the compost process. This can also lead to really strong odors and a loss of nutrients. We also tested for water content or moisture. So most of the macro and microorganisms, they actually live in a thin film of water within the pile. Too much water and the pile becomes saturated and there isn't enough oxygen for the organisms to survive. Too little water, this also limits the amount of organisms able to live in that habitat, and both can slow the compost process. So you really want that perfect amount that isn't oversaturated and isn't too dry. Oxygen is another important element. Many of the micro and macroorganisms responsible for composting are aerobic, and that just means that they require oxygen to survive. And although we didn't test for oxygen levels, we did account for airflow throughout the pile, which we will get into a little bit later. But this is why we often recommend to our cooperators to turn piles, especially at the beginning of the compost process, or use a forced air composting system that can blow oxygen into the piles. That way the microorganisms are getting the oxygen that they need to be able to function. And finally, we measured the pile for temperature. So temperature is what lets us know that the compost is occurring. Throughout the process, micro and macroorganisms generate heat, and this heat is what is able to eliminate harmful bacteria and weed seeds from your pile. As you can see from this graph, this really starts to occur right about 140 degrees Fahrenheit or 60 degrees Celsius. A compost pile has to reach, about 135 degrees to kill most pathogens and 140 degrees to kill most weed seeds. And it has to be able to sustain that for at least 3 days. So us measuring temperature throughout the composting process for all of the piles lets us know, how the composting is going and whether or not it's occurring, and really can be an indicator if there's something wrong or missing from the elements needed for it to compost correctly. So I talked a little bit earlier about the nutrients we had measured for this project. The goal for many farmers is to have a final compost product that's rich in nutrients that are essential for plant growth. All of the nutrients selected for this project already are naturally occurring in manure, and some of them are highly water soluble. So that means they can dissolve very easily in water and be transported by stormwater away from the pile. So nitrogen, that comes in different forms, and the levels of these forms of nitrogen change as the composting process occurs through a process called nitrification. The form of nitrogen most farmers are interested in is nitrates, which are essential in producing chlorophyll. They allow for fast plant growth, and they also are highly water soluble. Phosphorus was another important nutrient, essential in root development and plant growth. And potassium is also essential in plant growth, uptake of water, and root development. We also wanted to test for these nutrients because when they are leached from the pile during stormwater events and carried to our surface and groundwater, they can pose serious health issues for both humans and wildlife. These nutrients aren't just great for plant growth in fields. They also can cause an excess of plant growth in our rivers and our lakes and oceans. And this overgrowth of plants can cause a decrease in dissolved oxygen, which is critical for fish and other aquatic species to be able to survive. Additionally, nitrate poisoning in our groundwaters have been linked with a lot of human diseases, particularly in young children. And because of this, we wanted to understand really how effective each of these different dry manure storage practices are at limiting the amount of nutrients being leached into the environment. And finally, we also looked at the amount of fecal coliform that was leaching from the pile and that was being retained in the compost product. So fecal coliform is a group of bacteria. It lives in the intestines of all warm-blooded mammals. So humans and livestock alike both have fecal coliform, and it is present in warm-blooded animal waste like manure. Most fecal coliform is not pathogenic. So you can see in this kind of mid-color fecal coliform bubble, not all of the fecal coliform is harmful to humans or other wildlife, but a certain subset is. And so it is very expensive to test for individual pathogens or harmful bacteria like E. coli. And so we test for just fecal coliform as a whole, knowing that a subset of that bacteria is harmful bacteria and is pathogens and indicates pathogens in our surface and groundwater. So this map is showing the study sites where we performed some of our stormwater leachate sampling. So the purple outline that you see is a Unicoi Plateau Agricultural Production District, and the study sites are represented by the orange stars. And we chose the Unicoi Plateau area because there is a high density of livestock owners, and there are also several priority drainage basins nearby, the Newaukum Creek and Boise Creek. These tributaries contain critical spawning areas for threatened and endangered aquatic species. They currently have known resource concerns. They exceed Washington state standards for recommended levels of stream temperatures, dissolved oxygen, and harmful bacteria for salmon spawning, incubation, and juvenile rearing. And this pollution also has implications for human health concerns. We also had a third study site at the King County Farm in Maple Valley. The reason we did not have it on the plateau is because this is where we did our soil testing. And the Unicoi Plateau has a pretty unique geologic feature. The Osceola Lahar, it's a mud flow that came about from a series of volcanic explosions on Mount Rainier about 5,500 years ago. And it really created all of that sediment and debris when the mud flow happened during the eruption that's outlined in green. It created a very shallow hard pan throughout the plateau. And so that really interfered when we first started to do our soil testing there. And so King County was gracious enough to allow us to do some testing on their property, and that was in Maple Valley. So we worked with a steering committee of local farmers to pick the 4 most commonly observed manure storage practices. These practices also can be installed at different price points. So going from right to left, the first one, we call this the do nothing option. It's something most of us have seen. It's just a pile of manure on bare soil, uncovered. The second is a manure pile on the ground or soil, and that is covered with a tarp on it. And this would be a relatively cost-effective practice for most producers to implement. The third is a concrete pad that is uncovered, so no tarp or roof. And the fourth is the gold standard I mentioned earlier. So that is a concrete pad with a roof covering it. And this would also be the most expensive to install. I do want to note something unusual that you may note about these is that we do have walls in front of the manure bins and around the piles. This isn't something you would normally see. The reason for that is because we had to account for the volume of the manure to do analysis of this data later on. And I also wanted to note that we did account for oxygen that's needed for composting to occur by using a perforated drain pipe. And so that actually coils through each one of these manure storage practices. And then you can kind of see on some of these the tops of those coming over. And so that allowed for passive airflow and oxygen exchange throughout the composting process. So I wanted to show this photo. This is a covered and uncovered slab treatments before we put the manure in. And the purpose of this slide is to show how we collected the leachate. So each of these bins is tilted backwards at about a 2-degree angle, and you normally don't have pipes or holes in the back of your manure bin. You do want to keep them solid to allow all of the leachate to be captured, but we wanted to track what was coming off of these piles. So we added some pipes to the back, and that allowed us to transport whatever leachate was leaving the piles. And these went out into these pipes in the back and were gathered by a stormwater sample collecting machine. This is called an ISCO. And during a stormwater event, the ISCO would collect leachate samples periodically, and we would take a composite of those samples to the lab to be studied. Only the uncovered side had this machine because of the amount of rainfall and saturation. It had just a lot more runoff and leachate coming off of the pile. We were actually able to collect most of the leachate from the covered pile in rubber bins. So we didn't need to take individual samples during the runoff, which I think it was hundreds of gallons would come out during a season. And Zen could speak more to that. And so this is just showing you what leachate looks like as it comes off of a pile. I think it kind of looks like a really strong cold brew iced coffee. It's a little gross, but this is what we would take to the labs to be sampled, just to get a visual of what that might look like. And so for the covered and uncovered piles on soil, we would take 2 different sample depths or soil samples from depths of 0 to 12 inches and 12 to 24 inches. And these were all directly underneath the manure piles themselves. So to your left, you have an uncovered pile, and to your right, you have a pile covered with a tarp. And the purpose of taking those at different depths was to understand how these nutrients and pathogens were moving throughout the soil during or after stormwater events and throughout the season. And we took this periodically over a 4-month period. We also sampled the manure for all 4 of the different dry manure storage practice types. And this was to see how the composition of the manure was changing in relation to the nutrients and pathogens that we were interested in measuring. So to see what was being retained and what was being removed from the pile. And we would collect these samples periodically to have them analyzed by the lab. And as I mentioned earlier, we would also take temperature to monitor how the piles were composting over time. What's pictured here is a manure thermometer. It's something we recommend to a lot of our cooperators. It's a great way for you to understand at what point in the process your compost is at and kind of keep tabs on it and get an idea of whether or not you might need to add water, or maybe it has too much nitrogen or carbon. This can kind of help you understand, is it my composting process too slow? Is it getting a little bit too hot? And they're relatively cost effective. So we did use this thermometer at all 4 of the different practice types, but during our second year, we were able to get continuous monitoring of temperature through specialized equipment, which is really great. It just took the temperature continually over that entire 4 months, so we could see in real time, how that composting process was going, with much more granularity. And this was just for the sites that were on slabs, covered and uncovered. We were unfortunately not able to get them onto the treatments that were on bare soil. Okay. Did anybody have any questions? I'm going to stop sharing so I can see people.
Laura, we didn't have any questions go in the chat, but if you do have a question now, feel free to unmute yourself or just put it in the chat if that's easier. And if we don't have any questions right now, we can move on to Zen's portion. And feel free to keep putting any questions that do come up into the chat, and we'll get to them at the end of Zen's presentation.
Okay. Well, thank you, everybody. Great intro, Laura. Thank you so much. And so now I get the fun part of talking about the data and what we found. And so before I go into it, I just want to kind of reiterate the specific questions that we're interested in answering as it relates to placing the manure piles on a concrete pad or some other impervious surface area and putting a tarp on them. How do those two things affect the ability of the manure to compost effectively? How does that help with nutrient retention and therefore value retention to the producer? And then also, how does it aid in environmental protection? So as Laura mentioned, there are a few key factors for successful composting that we needed to make sure were in effect. We needed to make the piles big enough so that they were able to actually develop enough heat to get hot enough in order to get rid of the fecal coliform bacteria and make it safe to then apply to the land. And we also need to make sure that the manure material going in the beginning had that appropriate 30-to-1-ish carbon to nitrogen ratio. The carbon, again, is kind of the energy source. And then as the bacteria is feeding on that and assimilating the nitrogen, and then goes through that process of nitrification, turning the ammonia into the nitrate that the plants can actually use. And so over the composting period, as the bacteria use up that carbon, that ratio changes a little bit. And so the and so by the time it's done composting, the carbon to nitrogen ratio is down to around 10 to 15 to 1. The other two main components that we were monitoring was the aeration. And again, we used a passive aeration system for this with the perforated drainage pipe that was coiled, and we had it 1/3 of the way up and 2/3 of the way up, and then draped over the top to create a passive airflow system. There are active ways of doing this, either just by turning the pile physically or by setting up a blower system with similar kind of piping. But we wanted to decrease as many variables as possible, and so this allowed a low maintenance approach to basically just create the pile and then let it sit through the entire composting process without disturbing it, to minimize variation in the data we were collecting. The other main component is the moisture. So ideally, you want to start out between a 40 to 60% water content in the manure in order for it to effectively compost. And obviously, there's going to be a difference that gets affected for the uncovered versus the covered piles because the uncovered side is getting rained on. So that's one of the things that we wanted to look at in terms of how that precipitation was going to affect the composting of the uncovered versus the covered side. When it comes to keeping the nutrients and value in the pile, as Laura mentioned, there's different solubility factors for the different nutrients we're looking at. So primarily we're looking at the total nitrogen, which includes all the different types of nitrogen, the ammonia, the nitrate, and then the organic nitrogen as well. And also the total phosphorus and the fecal coliform bacteria. Those tend to be more sediment bound and organic matter bound. And so they don't dissolve as easily, they don't move through the pile as easily, and so they have what we call less mobility. However, the ammonia, the nitrate, and the potassium are highly soluble. And so they're going to be more impacted by a precipitation event, and they're going to move more quickly and easily through the pile if the pile gets rained on. So the basic kind of rule of thumb is if you smell it, that means it's volatilizing into the air, right? Volatilizing. If the ground is wet and the manure is sitting on the ground, it's leaching into the ground. And if it's if it's not going into ground, then it's running off, right? So those are kind of our three main pathways. And again, this is going to be the highest risk for the more soluble nutrients. So I'm going to start out by kind of giving the information in, I don't know, kind of a story fashion, was kind of the way I like to see it, is so we're going to start with the rain, and then we're going to track that water from the rain down onto the manure pile, see what it does to the manure. Then we're going to see the leachate that comes out of the manure pile, and then we're going to see how that leachate goes into the soil. So we're going to track the water through the whole system. So in the beginning, we have this is a precipitation record for the last 2 years of the study. And the first year was, compared to the 30-year average, it was a bit drier, especially during the winter. We ended up having fewer samples than we anticipated. And the second year, it was a wetter year. So there's natural variability in both seasons that we did the project. We actually ended up combining the data from both seasons for a single analysis, just so that we had a more robust data set to minimize variability and kind of pull out the trends more easily. And so one of the first questions is it composting? And this is the real-time temperature data that Laura mentioned. And so here's an example from our 2 slab sites with the red color being covered and the yellow being uncovered. And you can see that the covered reaches a higher temperature on site H and maintains that higher temperature throughout the entire composting, which is kind of what we expected, because when a pile gets rained on, it tends to cool the pile down. Now at site A, we were monitoring and we didn't see the same difference in the temperatures. And when we got our first round of manure samples back from the lab, we knew why. It was because one of our key components, the moisture percentage of the pile, was not enough to get the composting to the level that it needed to be. So we had to spray extra water on the pile, basically. And once we did that, that kind of kicked the composting into high gear again, and it was able to then maintain additional high temperatures compared to the uncovered side for the rest of the study. But here's a prime example of, you don't know if it's not working unless you have the data to monitor it. And so that was why we were tracking this, to make sure that it was working correctly. So because we know that the high temperatures is what is going to kill off the fecal coliform bacteria, and we got high temperatures, the question is it safe to apply? And so we did measure the fecal coliform counts in the manure piles, and we saw a very rapid drop off. Within the first month, 2 months, it was pretty much all gone. And so that's what we'd expect from a successful compost. And this was actually shown to be effective in both the covered and the uncovered piles. So that means you're free to spread, right? Well, not quite. We still want to make sure that the nutrients that you're applying are going to stay on your land. And so I just want to take a quick moment to let you know that there is a tool that Whatcom Conservation District developed called the Application Risk Management Tool. And so if you just go to whatcomcd.org/ARM, it's a free tool. It's just a map online, and it shows you runoff risk assessment. It was designed for dairies spreading liquid manure, but it's useful for anyone who's going to be spreading nutrients. So whether that's compost or fertilizer, it will let you know based on recent weather and soil saturation, and it actually is tied into the NOAA 3-day forecast. So if it's expected to rain in the next few days, it takes all that into account, and then it lets you know red is high risk, no go, and green is good to go. And so I just recommend that before you land apply your compost, to make sure that the nutrients stay where you want it to be, go ahead and check that before you apply. So now let's come back. So the rain came through, and we know that the temperatures got high enough to kill out the fecal coliform bacteria. So now let's look at the nutrients in the manure pile. So we're going to start with the low solubility nutrients. Remember, the total nitrogen and the total phosphorus are largely sediment bound. And so we didn't see a very big effect throughout the study with those two. If you look at the total nitrogen by April, seems to be a sizable gap there. And we're going to explain that in a little bit. And that's because of the soluble components that go into that total nitrogen. So now let's look at that. The higher solubility, the ammonium, the nitrate, the potassium, these are the ones that are more susceptible. These are the ones that you'll lose from your pile if you don't cover it. And so the nitrification process, remember, the microbes transform the ammonium into the nitrate, which is what the plants can actually use. And so during that composting process, during that transformation process, you can see that the covered side, the kind of burnt orange color, continues to increase over time, whereas the uncovered side, if anything, went down a little bit. So what that means is if you put a cover on it, you're keeping the nitrate as it's being produced in the pile. And if you don't cover it, it's getting washed out. Potassium showed the same overall trend. It's also highly soluble, and so it's susceptible to leaching if it isn't covered. And we saw the same thing with the higher potassium levels being maintained in the covered bins, whereas they were getting washed out in the uncovered bins. So what does this really mean then? Well, this means that as a farmer, if you start out with your manure pile, you have a choice. You can either put it in the covered bin or the uncovered bin, right? You can put a tarp on it or not. It's a very clear difference in the outcome that you're going to get. As the manure decreases in either scenario, our study shows that if you cover it, the nitrate actually increases by 9-fold, the 900%. Whereas if you don't cover it, you actually lose 80 or 32% of your nitrate. So that is what you want to apply to your fields. This is what the plants want to eat, right? And you're losing all of your nitrate if you're not putting the top on. And the same thing happened to the potassium as well. You either keep all the potassium if you cover it, and you lose pretty much all of it if you don't. It's pretty straightforward. So now we know what happens to the pile if you don't cover it. Let's look at that leachate. We have an idea now of what's coming off, but let's actually look at the data. So the fecal coliform data, notice this is a log scale. Oops, I'll come back. This is a log scale. So this isn't kind of an exponential drop off. But once you put it in this scale, it looks pretty even. It comes down, and both the covered and uncovered are pretty similar. And that's because the fecal coliform is basically being eradicated before it leaves the manure pile. So there isn't a lot to leach or not leach out. The total nitrogen, we're also seeing kind of an increase overall in the uncovered, which just means that there's more nitrogen coming out of the uncovered pile, as well as now we look at the, and the total phosphorus is the same thing. There's more phosphorus coming out of the uncovered pile. And the nitrates, again, even more susceptible. So there's even more coming out of the uncovered pile. But the thing I want to point out here is when we calculate the amount of nutrients coming off of a pile, we're not as much interested in just the concentration, which this is showing. We also need to know the volume that's coming off, because it's the concentration times the volume that then tells you the load or the overall amount that's coming off. And so if I look at this slide, there's variation, but it's in the same, I mean, it's comparable, right? It's in the same factor, so to speak. However, that's not true with the volume. When I look at the volume that came off the covered side versus the uncovered side, our covered side had on average 31 gallons on the slab site that came off per year. And the uncovered is 622. So that's a factor of 20. So there's 20 times. So if you imagine it's the same concentration, so basically the same leachate coming off of each side, but you got 20 times the amount coming off of that uncovered side, it starts to put things into perspective. And then when we actually do that calculation and we look at what this means, again, it stands out in massive factors. The fecal coliform is the same because there isn't much left in the pile to begin with. But once we start looking at the nitrogen and the phosphorus, then there's huge differences. And that's a combination of the extra volume and the concentrations coming off the uncovered pile. So to a farmer, the whole point of doing that compost is that you want to be able to apply it to your fields. You want to be able to use it as a fertilizer for your plants. And what the data is showing us is if you can go through the motions of composting, you can even conduct successful composting and drop the fecal coliform count and make it safe to apply. But if you don't put a tarp on it, you're losing the very nutrients that you want to apply to your land. This nitrate 27X, what this means is that 27 times the nitrate that came off of the covered side came off the uncovered side. So now that we know it comes out of the manure pile, it comes off in this form of the leachate. What if you don't have it on a pad? What if it's just sitting on the soil? How does that affect how those nutrients go into our soil? So now we're looking at the soil concentration. Remember, those boxes we had sitting directly on the soil, and we were sampling at 2 different depths. So they would, the staff would stand outside the box and sample at an angle to go underneath the pile to collect these samples. So we're getting soil that's directly beneath the pile without disturbing the pile. And so if we look at total nitrogen, for instance, this burnt orange is the 0 to 12 inch on the covered, and the 0 to 12 inch on the uncovered is kind of this salmon color. And they start out, and the 0 to 12 and also the 12 to 24, they start out even. That's great. So now we can see what kind of difference it makes going forward. And by the time we get to January, there is an increase in the total nitrogen in the first foot, but there's a lot more in the uncovered pile. And we know that has partly to do with the fact that there's a lot more volume from the rain on the pile, pushing those nutrients into the soil, percolating into the soil, infiltrating into the soil. Phosphorus saw not as much of an impact. There is a little bit by the time you get to January. You can tell that there's definitely an increased amount in the uncovered pile, but the phosphorus wasn't impacted as dramatically as the nitrogen was. The fecal coliform is an interesting thing because at first we weren't quite sure what to make of this. When we saw the fecal coliform moved into the soil, as we might expect, but then in January, it all disappeared. And so through some additional research, what we realize we think is happening is that at that time of the year, the temperature of the soil in that first 2 feet of the ground is actually causing the fecal coliform to go dormant. And so then when the lab was analyzing the samples and the colonies weren't growing, it came back as low concentrations, when in fact, they could still be there. They're just dormant, and then they would reappear in the spring, which is what we tend to see in a lot of areas around the Puget Sound region. And as you'd expect, probably no surprise, the high solubility nutrients, the ones that move more easily through the pile, also move more easily through the soil. So the ammonium, the nitrate, and the potassium all showed strong trends, with the uncovered pile having, not just in the first foot, but in the second foot as well, higher concentrations moving down through the soil over time. Again, I rarely get to see data that is this cut and dry. This is as straightforward as it gets. You put a tarp on it, you keep the nutrients in the pile. If you don't, they're going to leave the pile. And if it's on soil, it's going into the soil. So that's basically our key takeaways. Cover the poo. Covering the piles is going to reduce the nutrient and the value loss to the producers due to precipitation. And it's going to allow the pile to maintain higher temperatures and be more effective and increase the rate at which it composts. And if you don't have the money for a bin, and you just need to stick it somewhere on the ground, then at least put a tarp over it, because it's a very low cost option that's going to make a big difference in the amount of leaching that actually occurs off of that pile. All right. So any questions?
Question, we have pasture donkeys and try to collect manure from the field weekly. Is there a best practice in terms of just leaving it in the field and spreading with a harrow, or collecting and composting? Great question, Diana. Thank you.
Thanks, Diana. So typically when we collect manure or talk about collecting manure, it's during a certain time of year, and that's a rain season. And that's when we recommend that folks that have livestock put them back into a confinement area or a heavy use area. And this is really to protect your fields from compaction. So the weight of the livestock on the field, that can really damage your soil. It can damage the plants when they try to grow back. But it also, helps with animal health to not be standing around in potentially muddy water. So just keeping them off of wet fields. But in order to compost and get rid of those pathogens, retain those nutrients, Zen had mentioned you need a pile of at least a certain size. And that's 3 feet by 3 feet. So if, every single operation is different, if you live in King County or I'm not quite sure where you live, but most counties do have a conservation district, and they do have technical planning services, and they can come out and look at your operation, what your management goals are, and really come up with a plan that can help you decide what the best course is for manure management, that's specific to your property, your animals, and your goals. So not really knowing what your farm looks like or your whole setup, I would say that you're not going to be able to compost that manure or retain many nutrients leaving it out there. But that we really are talking more of like a wintertime thing, or whenever animals are confined and you can get out there. And in general, we recommend picking up every day from those confinement areas, because that organic material over time can really create a muddy situation in there pretty fast.
Great. Thanks, Laura. Thank you so much, Zen and Laura, for your great presentation. Thank you for Nick with your help on Zoom today. And thank you to all of our attendees for joining us.